Good evening. My name is Kristina, and I'm doing my presentation on biotinidase deficiency. So, an overview of biotinidase deficiency. This is a genetic condition that prevents the body from recycling the nutrient biotin. Signs and symptoms of this disorder usually show within a first few months after birth. This disorder is caused by a mutation in the BTD gene that is responsible for making the biotinidase enzyme. This enzyme is responsible for breaking down or cleaving biotin that is attached to proteins in food, leaving biotin free or in an unbalanced state. Having a defective biotinidase means that biotin is unable to be released, creating a biotin deficiency in the body. Biotin is defined as a B vitamin present in foods such as liver, egg yolks, and milk. Biotin-dependent carboxylase enzymes require free biotin to break down lipids, proteins, and carbohydrates in food. Other names of this condition are called BIOT, BTD deficiency, carboxylase deficiency, and many others. Physical effects and symptoms. So biotinidase is an inherited disorder, and if not caught early, can result in more severe symptoms. There are two types of biotinidase, a more severe version and a milder version. So profound biotinidase, which is the more severe version, includes symptoms such as seizures, hypotonia, breathing problems, hearing and vision loss, difficulty with movement and balance, skin rashes, hair loss, and a fungal infection. Children may also experience delayed development as a result of the deficiency. The more milder version, which is called partial biotinidase, has symptoms such as hypotonia, skin rashes, and hair loss. These symptoms may also only arise during periods of illness, infection, or times when the child's body is under stress. These symptoms and complications can be avoided or even improved if they start a long-term treatment right away. Prevalence and frequency of biotinidase deficiency. Biotinidase deficiency, profound and partial, occurs in approximately 1 in 60,000 newborns in the world. This deficiency is more common in individuals with European descent and also Turkish, Saudi Arabian, and Japanese descent. The responsible gene and location of this gene. So the responsible gene for this genetic disorder is the BTD gene. Mutations in this gene reduce or eliminate the activity and availability of the enzyme biotinidase in the body. This enzyme is really important because it breaks up and cleaves the complex, so biotin is now free and can be reused by other carboxylase enzymes. The biotinidase gene is located on chromosome 3p25 and consists of 4 exons with a total length of 1,629 base pairs. So the type of mutations that result in biotinidase deficiency are all types of mutations. So base substitutions, deletions, and insertions all can result in biotinidase deficiency disorder. A mutation in the BTD gene reduces or eliminates the activity of biotinidase, resulting in either profound or partial biotinidase. Profound biotinidase is when the enzyme's activity is reduced to less than 10% of its normal activity. Partial biotinidase is when activity is reduced to between 10 to 30% of its normal activity. Without enough of biotinidase enzyme, biotin cannot be recycled, which can impair the activity of biotin-dependent carboxylases, leading to buildup of potentially harmful toxic compounds in the body. If the condition is not treated promptly, this buildup can lead to damage in various cells and tissues, and children can experience hypertonia, rashes, hair loss, and a variety of other symptoms. Mode of inheritance. So this disorder is inherited in an autosomal recessive fashion, meaning that both copies of the BTD gene in each cell are mutated, with 2 lowercase letters. The parents of a child with biotinidase deficiency will each have 1 copy of the mutated gene, A, and 1 normal gene, capital A. If, as you can see to the right on the Punnett square, this is the cross between 2 parents who are carriers, unaffected carriers of biotinidase deficiency. So when you cross the 2, there is a 25% chance that the child will be normal and not have biotinidase deficiency. There is a 50% chance that they will be a carrier, but they will not be affected. And there is a 25% chance that the child will be affected by this disorder and have biotinidase deficiency. Simple pedigree inheritance pattern. So here is a simple pedigree that was drawn up showing 4 generations of a family showing the biotinidase deficiency disorder. So the first generation is between John and Sally. John is a carrier of biotinidase deficiency. So it would be capital A, lowercase a. Sally is biotinidase deficient, as shown by the little red triangle in her shape. So her letters would be lowercase a, lowercase a. When crossed, they had 4 children: Kristina, Matt, Logan, and Steve, which represents the second generation. And as you can see, that 2 out of the 4 are carriers of biotinidase deficiency, and the other 2 out of the 4 are biotinidase deficient. You can follow through the next generation, the generation after that, when they have kids, and just think about from one generation to the next doing a Punnett square between the 2 parents. So between a carrier parent, one who's unaffected, one who has biotinidase deficiency, and going through and seeing the probability of their children being affected or not affected. The genetic testing options that are available to diagnose biotinidase deficiency include newborn screening, target mutational analysis, and a full gene sequence. Newborn screening is a blood test, which is a few drops of blood taken from the heel of a baby, and this occurs about a few weeks after birth before symptoms start to appear. This is required for all newborns to check for any underlying conditions to start early treatment. It's important to note that not every country checks for the BTD mutation during newborn screening process, but it is something that we look for in the US. If abnormal results appear on the panel, a diagnostic workup is ordered. This can include quantitative biotinidase activity and DNA testing for specific mutations, which should be included in the diagnostic workup. Types of genetic testing include targeted mutational analysis, which identifies common variants causing partial or total deficiency of biotinidase, or full gene sequencing, which is much more common. Biotinidase cannot be detected through amniocentesis. It's not something that is tested for while the child is still in the womb. If parents are concerned that they're carriers and want to know the likelihood that their child will develop it, they should talk to a genetic counselor or their general practitioner for the next step. Is genetic testing for this optional or mandatory? Newborn blood work is required for all children a few weeks after being born to see if the child has a condition, or further diagnostic testing may be asked or required. Further diagnostic testing is required if the child has an out-of-range screening. Genetic testing may be out of order to see if there is a BTD gene mutation. As you can see in this picture, this is a newborn and they have their little heel pricked, and this is what a newborn screening little panel would look like. The blood drops from the heel. Importance of early detection and treatment. So early diagnosis and treatment of BTD is extremely important. This can prevent symptoms such as seizures, hypotonia, ataxia, developmental delays, and problems with hearing and vision from happening. If it is detected early, it can be treated early with oral biotin before symptoms start to appear. So as you can see in the picture, in the lower corner, this is a baby who has biotinidase deficiency and is taking oral biotin as treatment for his symptoms. If you or someone you know is concerned about passing a genetic disorder down to the next generation, speak to your general practitioner or a genetic counselor to talk about the option of genetic testing. Or if you're just curious, you can look into the genetic testing options. And here are my references that I used for my presentation. And thank you for listening to my presentation. I hope you learned something about biotinidase deficiency and genetic testing.